Saturday, I told you which we did in the process of transcription. Now, I start with the process. Now, what happens in the process is, first thing, activation of ribonucleotides. During the process, the first step is activation of ribonucleotides. Now, what happens during the process of activation of ribonucleotides? The inactive form of ribonucleotides, which is AMP, adenosine monophosphate like AMP. We have others also, guanosine monophosphate. Then we have cytosine monophosphate. We have here, in this case, we will not have TMP because it is what is formed is a messenger RNA has to be formed by the process of transcription. So in RNA, we do not have T, we have U. So we will have here UMP, which will get activated. Then this, in the presence of enzyme, the enzyme relays, they are these inactive form of energy. These inactive forms of energy in the presence of enzyme phosphorylase gets converted into active form, and that active form will be ATP, GTP, CTP, and the next one is UP. So these are the active forms which are formed from the inactive form. Now, after this, what happens? In the next step, this is DNA template is identified. So DNA template is identified. During this process, what happens? In the transcription unit, which was like this, template strand and the coding strand, the promoter site is at here, and the terminator site is here. Okay, we have this. We have 3' and 5', 5', 3' here like this. So this is identified. How it is identified? Because this promoter is here on the 5' end of the coding strand. So this presence of presence of promoter is identified, presence of promoter as well as the presence of template strand. This is the template strand. This the top strand is the template strand. This is identified. And after the identification of this strand, what happens? Here the RNA polymerase gets recognized, or by the help of sigma factor, RNA polymerase recognizes it. And in case of eukaryotic, it's only the RNA polymerase which recognizes and gets attached. So it gets attached to it, and then what happens? It moves along. It starts moving along this. Identifies the template strand. So the third step was initiation of the formation of messenger RNA. So this is initiated. The RNA polymerase which gets attached to this promoter site or it can be a rho factor, sigma factor in case of eukaryotes, through sigma factor in case of prokaryotes, and just simply by attachment of formation of messenger RNA starts. And here DNA template is identified by RNA polymerase. RNA polymerase attaches itself and recognizes. In case of prokaryotic cell, it is a sigma factor through which this RNA polymerase attaches to the promoter. And in case of eukaryotic cells, means the cells in which nucleus is well defined, it's only RNA which attaches to the this promoter site. And then what happens? It starts sliding along this template strand. When it starts sliding along this template strand, what happens? Initiation of the formation of messenger RNA takes place. This is on the basis of nitrogenous bases present on the template strand. That means the bases, nitrogenous bases, which are present on the template strand, on the basis of this, a new formation of a new messenger RNA is initiated. This is initiated. So this is known as initiation. After initiation starts the base pair. So what happens is the messenger RNA starts synthesizing. It starts synthesizing. How does it start synthesizing? It starts synthesizing using ATP, UTP, GTP, CTP, etc. So this messenger RNA gets synthesized by utilizing raw material which is there in the cytoplasm, in the nucleoplasm. This is taking place in the nucleoplasm of eukaryotic cell, because in case of eukaryotic cell, the cell is divided into nucleoplasm and cytoplasm. So this process of transcription in case of eukaryotic cell takes place in nucleoplasm. If this is cell, a eukaryotic cell, this is a defined nucleus. Whereas if this is a prokaryotic cell, the nucleus is not defined. The nuclear material lies like this. Defined nucleus is not there. That means nuclear membrane is absent. So the nuclear material lies like this. So there is, in case of eukaryotic cell, there is difference between nucleoplasm and cytoplasm. Whereas in this, it's one. So we will not say in this case that it is taking place in the nucleoplasm. It is taking place in the cell, we will say, in prokaryotic cell. Whereas in eukaryotic cell, we will say that the process of transcription takes place in the nucleoplasm. And then after the process of transcription, the messenger RNA which is formed, it comes out of the nucleoplasm into the cytoplasm, where the process of translation is. So now here what happens, the initiation of formation of messenger RNA starts utilizing what is utilized? The active form of energy. This is active form. The active form of energy and this active form of UTP form of energy is utilized, and this initiation takes place, and by the process of initiation this initiates, and finally we can use rather this as fourth point. This can be used as fourth point, base pairing. This can be used as fourth point, base pairing. During base pairing, ATP, GTP, CTP, UTP is utilized. This is utilized in the process of base pairing, and this takes place where? In the nucleoplasm, which takes place in of course the complete process in the nucleoplasm. And during this process, what happens? This ATP, all these forms, these are hydrolyzed, broken down. in the presence of enzyme pyrophosphatase. This we have already done to release energy. They get converted into their monophosphate form, first diphosphate and then finally monophosphate forms, and with the release of energy. Monophosphate forms plus energy is released. And energy is pyrophosphatase. The enzyme which is used is pyrophosphatase. And then as a result of this base pairing takes place. Base pairing in the sense we will not use here complementary arrangement is base arrangement because no pairing takes place here. The bases are arranged. Like here, on the basis of the bases which are present here, a new strand is formed, but there is no bonding between this and this. There is no bonding because this is a separate entity which is formed. This is not connected with the transcription unit. There is no bond formation between messenger RNA or the template strand of DNA. So we will not use the word like base pairing. This is the base arrangement which has taken place. And as a result of this for this formation of this messenger RNA, the energy is utilized by the hydrolysis of the triphosphate. When these triphosphates are converted into monophosphates with the release of energy. And then finally, what happens that after this formation of RNA, it continues. The chain is formed. Then formation of RNA, messenger RNA. Finally, the fifth point, synthesis of messenger RNA. It takes place when this is completed. When complete messenger RNA is formed, then what will happen? This is in the form of a chain. This is in the form of a chain. Messenger RNA is formed. And this is the messenger RNA which is formed on the basis of the bases which are present here. So this is formed and finally chain separation takes place. The next step which takes place here is chain separation. Chain separation. Here the word chain is used for messenger RNA. The word chain, this is used for messenger RNA. That means the messenger RNA which is formed here now, you know, it keeps on forming. See what will happen from here. The RNA polymerase will keep forming and will keep moving until the RNA polymerase reaches here. Long chain is formed. A long chain of messenger RNA is formed. This long chain of messenger RNA now, which is formed completely, needs to be removed. So now that means it has to be separated from this transcription unit. How this chain of messenger RNA is separated from this transcription unit? This is separated in case of in case of prokaryotes and eukaryotes in a different way. If it is a prokaryotic cell, prokaryote means say a bacterial cell. If it is a prokaryotic cell, in that case, if this is taking place in a prokaryotic cell or in a bacterial cell, then in that case, what happens? That a new factor. See, in case of prokaryotic cell, this process of DNA messenger RNA formation started with sigma factor. Same way, in case of prokaryotic cell, this formation of messenger RNA will also end up with another factor, and that factor which is used here is Rho factor. Rho factor, in words we call it Rho, RHO, Rho factor. So Rho factor helps in this. The Rho factor in case of prokaryotic cells attach here, and then finally it helps in the separation of this chain. So what is three things are separated? Then one, we get messenger RNA. Rho factor itself gets separated. Rho factor, after separating this, gets separated, and RNA polymerase. So three things we get after separation. So here, in case of, if it is the prokaryotic cell, it's the Rho factor which helps in separation of the chain. But what happens in case of the eukaryotic cell? In case of eukaryotic cell, this is done by some, segments which do not code for any amino acid, the nonsense codon. If it is a eukaryotic cell, then, or we can say introns. Which introns? Specifically here, the stop codons, also known as the nonsense codons, which help in separation. That means here one nonsense codon is formed which do not code for any amino acid, and here it separates. So messenger RNA gets separated at that point, and after this, finally, what we get, the messenger RNA, which is, unrefined, crude messenger RNA, which is not properly formed messenger RNA, which is not a mature messenger RNA, which is heterogeneous RNA, what we get. So after this, what we get is, after this separation, in case of eukaryotic cell, if it is a eukaryotic cell, and if it is a prokaryotic cell, we will get different types of RNAs in both. If it is a prokaryotic cell or bacterial cell, if it is a prokaryotic cell, I'm writing just PROK. If it is a prokaryotic cell, in this we will get a complete strand of messenger RNA. A complete strand of messenger RNA with all, sense codons, which will code for the amino acids. That means no, we use two terms. Before also we have done, we used the terms exons and introns, INTRON. If you remember, we use the two terms exons and introns. Exons have codons which code for amino acids. So they have codons coding for amino acids. But introns do not have. They do not have codons which code for amino acid. They have all nonsense codons, all codons which do not code for any amino acid. So what I am telling you, that in case of prokaryotic cell, if it is a prokaryotic cell, the messenger RNA formed will not have any introns. It will have all exons. No introns are it was a little interruption, like the heterogeneous RNA which is formed. Even I have interruptions of introns in between. So here, as I have made here the diagram, in case of if it is a eukaryotic cell, the messenger RNA which is formed. This is the messenger RNA which is formed, and this messenger RNA, in case of eukaryotic cell, is not continuous. It will have some interruptions, and those interruptions will be in the form of introns. So if we consider this to be a coding sequence, so this is exon, which is a coding sequence. This is intron. Then again we have exon, then intron, then exon, then intron. So this way we will have interrupted sequence, and this RNA is known as heterogeneous RNA. The messenger RNA which is formed, it is still not processed. This is unprocessed, and this is heterogeneous RNA which we get as a result of transcription in eukaryotic cell. And finally, after this, what happens? This is prepared. Now this can go for translation. This, in case of prokaryotic cell, this is process. This is what is required for the process of translation. So this messenger RNA, in case of prokaryotic cells, will go for the process of translation. But in case of eukaryotic cells, this is still unprocessed, and this cannot, just a minute. This cannot go for the process of translation. So it has to be processed now. And that process by which it is processed, or this is converted into a usable form of messenger RNA, or this we will not call messenger RNA, which I have written here. This is known as hnRNA, which is heterogeneous RNA, because this has the uncoding sequences also, which are introns. Now this has to undergo the next step, which is seventh step, and which is only in case of eukaryotic cell, not in prokaryotic cell. Because in case of prokaryotic cell, we have got a messenger RNA which do not have any uncoding sequence, which has all the coding sequence, which do not have any introns, which has all the exons which code for amino acids. So it is a ready form of messenger RNA which can go to the next step of translation and then further synthesize protein. But in case of eukaryotic cell, what we have got as a result of transcription is not a true messenger RNA. It is heterogeneous RNA. It is not messenger RNA. It is heterogeneous RNA. That means it has coding sequences as well as uncoding sequences. See, these exons are coding sequences. These are uncoding sequences. You know what are coding and uncoding sequences? Suppose we have U. This is a coding sequence because this U, it codes for phenylalanine. Phenylalanine. This is an amino acid. What is this phenylalanine? This is an amino acid. So when any codon, when any triplet codon, why triplet codon? Because it has 1, 2, 3 bases. When any triplet codon codes for an amino acid, we say that it has a coding sequence, or it is an exon. But when it does not code for any amino acid, we say that it does not code for any amino acid. This is not coding for amino acid, or that is an intron. Okay, so here this what we get as a result of transcription in case of eukaryotic cell is heterogeneous RNA, which is not a true RNA. It has coding as well as non-coding sequences. So now see what is used for the process of translation. See, after transcription we have translation, and what is required for the process of translation is only exons. These introns which do not code for any amino acids are not required, so these are to be removed. We do not need introns. We do not need these introns because they do not code for any amino acid. They do not code for any amino acids, so we do not need them. We need only exons. So this is done. This has to be done now to this hnRNA, which is formed as a result of transcription. That means from this hnRNA, these introns needs to be removed. And what is that process of removal of introns? That process of removal of intron is known as splicing. So this we will write under post-transcriptional changes. Post-transcriptional changes means these are the processes which takes place after transcription. Post means after transcription, whatever changes takes place, these are post-transcriptional changes. So what happens in post-transcriptional changes? In post-transcriptional changes, in case of eukaryotic cells, these post-transcriptional changes are not observed in prokaryotic cells, not observed in prokaryotic cells. But if it is a eukaryotic cell, or in case of eukaryotic cells, it takes place. And what happens during this takes place? Splicing. Splicing takes place. And what is this splicing? Splicing is removal of these introns which are non-coding sequences. Splicing takes place, and this splicing is the removal of non-coding sequences. Removal of non-coding sequences, that is. And finally, when these non-coding red interruptions, when these are removed, what do we get? We get all introns together, and which is now messenger RNA ready to undergo the process of translation to synthesize. So this is now ready to undergo the process of translation to synthesize proteins. So this difference between the process of transcription in prokaryotes and eukaryotes. Along with this, we will do how many types of RNAs are formed. So this is all for today. I think the time is over. I end the meeting here.